All right, let's get straight into it. Today we're going to take a real AQA GCSE Biology exam question and completely break it down step by step. And our secret weapon? The official mark scheme. By the end of this, you are going to know exactly what the examiners are looking for and how to give it to them.
So, what's our game plan? First, we'll decode the question itself. Then we're going to tackle that big 6-marker. After that, we'll dive into handling data, master a pretty tricky graph calculation, and finally, apply our knowledge to explain the results. Let's do it.
Okay, let's get started. Our focus is question 4 from the June 2023 higher tier paper. And trust me, this is a fantastic example because it tests a whole range of your skills all in one place.
And here it is, in all its glory. Look at this thing. It starts with a 6-mark essay question on nutrient cycles, then it jumps straight into interpreting experimental data, plotting a graph, calculating a rate from that graph, and then finishing you off with a 3-mark explanation. It's a complete workout for your biology brain. Right, let's tackle the beast first. Question 4.1. It's asking how carbon and nitrogen from dead leaves get recycled. Now, 6 marks are up for grabs here, so the key is all about having a good structure and making sure you hit those very specific points from the mark scheme.
So the question itself basically gives you a massive clue. It mentions both carbon and nitrogen. So to get into that top mark band, what do you think you need to talk about? Yep, you absolutely have to address both the carbon cycle and the nitrogen cycle. And this is the important bit. Show how they're linked together through decay.
Okay, let's look at the carbon cycle first. The mark scheme is looking for a clear story. You get points for mentioning that microorganisms, bacteria or fungi, cause decay using enzymes. Then you get more marks for explaining that this process digests and breaks down large molecules. What do these microorganisms do next? They respire, releasing carbon dioxide. And where does that CO2 go? Into the air, where plants can then take it in through their stomata to use in photosynthesis, making things like glucose. See? Each of those steps is a potential mark.
Now for the nitrogen part. The mark scheme shows that the very same decay process also releases nitrate ions into the soil. Plants then absorb these nitrates through their roots, and, for an extra bit of detail, they do this using active transport. And why do they need them? Well, to build all the really important stuff like amino acids, which of course they use to make proteins.
So this is it. This is the crucial point, and it's a quote taken directly from the examiner's guidance. To get those top marks, you can't just talk about one cycle. You absolutely must describe the breakdown and the use of both carbon and nitrogen compounds. Linking the two is the real secret to a top-level answer.
All right, let's switch gears. The next part of the question moves on from pure theory and gets into your practical skills. We're now looking at an investigation into the decay of milk.
Question 4.2 asks for just one improvement to the method, and the mark scheme gives you this whole list of perfectly good answers. You could suggest using more temperatures to see the pattern better, or maybe measuring the pH more often. A really solid answer is always to repeat the experiment at each temperature and then calculate a mean. That always improves reliability. Any one of these will get you the mark.
Now, let's get to the graph in question 4.3. You're given the data for 25 degrees Celsius, and you're asked to plot it on these axes. This is a test of precision, pure and simple. Your first job is to get those 3 data points plotted in exactly the right spot.
And here are the rules, and they are strict. You get up to 2 marks just for plotting the points. That's 5.3, 4.9, and 4.8 correctly. They give you a tiny bit of wiggle room, plus or minus half a small square. But the 3rd mark is for the line of best fit. And notice what the mark scheme says. It must be a correct curved line. If you just join the dots with a ruler, you get 0 marks for that line. It has to be a smooth curve.
Okay, this next part, 4.4, is where a lot of students can lose marks. It's a 4-mark calculation, asking for a rate of change from a curve. But honestly, if we break it down using the mark scheme, it's perfectly manageable. So the very first step, and this is your first mark right here, is to draw a tangent to the curve. The question is very specific. It has to be on the 15-degree curve and at exactly the 2-day mark. Remember, a tangent is just a straight line that just touches the curve at that single point.
And this slide shows you exactly what a correct tangent should look like. Getting this line right is your first mark in the back. From here, everything else follows. You're going to use this straight line, not the curve, to work out the gradient.
Step 2 is calculating the gradient of your tangent. Just remember, gradient is rise over run, the change on the y-axis divided by the change on the x-axis. The mark scheme's example tangent shows a change in pH of 2 over a time period of 4 days.
That quick calculation gives a rate of 0.5, and that secures you the next 2 marks. Easy as that.
So for the final mark, we have to actually answer the question, how many times faster is the rate? Well, the question kindly tells us the rate at 5 degrees is 0.3. We just calculated the rate at 15 degrees is 0.5. So the final step is to divide our rate by the rate they gave us. And that calculation brings us to our final answer, 1.67. The mark scheme also notes that no unit is required, so that number is all you need for the final mark. Job done.
Okay, let's move to the final part of question 4. This is where it all builds up. Question 4.5 asks you to explain why the pH changes more quickly at higher temperatures. This is where you apply your core biology knowledge.
To get all 3 marks here, you just need to make 3 clear links. First, you state that at higher temperatures, enzymes are more active, or maybe the bacteria reproduce more quickly. Second, you explain that this causes lipids in the milk to be broken down faster. And third, you connect that breakdown of lipids to the faster production of fatty acids, which is what causes the pH to drop. Each of those points is worth a mark.
What's also really interesting is that the mark scheme allows for an alternative, equally valid explanation. You could have talked about particles having more kinetic energy at higher temperatures. This leads to more frequent successful collisions, and so faster production of fatty acids. It's a great reminder that there can be multiple correct ways to explain the science.
So, after deconstructing that entire question, what are the big takeaways? What have we learned about exam technique by going straight to the source, the mark scheme itself?
Well, we've seen exactly how to structure a long answer question by hitting all the key points for both cycles. We've learned to be super precise with data plotting and to follow the rules for lines of best fit. We've mastered the step-by-step process of calculating a rate from a curve using a tangent. And crucially, we've seen how to apply our knowledge and that sometimes there's more than one right answer. You see, the mark scheme isn't a secret document. It's your guide to exactly what's required. Using it is the smartest way to revise, period. So the only question left is, which question are you going to apply these skills to next?